Okay, hello everyone. Welcome to Concordia University's Fourth Space. Thank you for joining us for today's event, Art Meets Science, Storytelling and Research with Dr. Matteo Farinella, part of the SynBio Expressions series here at Fourth Space this week. Now to help situate you, we are streaming to YouTube, live from Fourth Space, and we are located on unceded Indigenous lands in Tiohtià:ke, Montreal. Here at Fourth Space, we work with our university community to help mobilize knowledge, co-creating daily activities that examine research questions and projects across the university. And we're running today's event as a live stream Zoom meeting, so anyone joining us there, just pop any questions or comments into the chat. That would be great. Those of you here in the space, we'll be moving some microphones around. If you have any questions or things like that, just let us know. With that, it's my pleasure to pass the microphones over to the organizers of this event series, Kathleen Hahn and Zoe Katz. Welcome back. Over to you.
Thank you, though. I guess the few first remarks we start with, we gather today in Tiohtià:ke, the ancestral and unceded lands under the custody of the Kanien'kehá:ka Nation. When we discuss the institution of science, it is especially important to consider the difficult history of science and indigenous knowledge systems and how it has shaped science methods. Science has, for a long time, attempted to distance itself from the context around it as to avoid bias, and in doing so has neglected to address anti-indigenous sentiments that have shaped its findings, its funding, and its use. More recently, initiatives featuring scientific collaborations with autochthonous groups have resulted in scientific projects that address real-world needs and incorporate indigenous and community knowledge. The work is long. We encourage you to reflect on how colonial roots have shaped science and the future avenues ahead. A commitment to democratization cannot be complete without a commitment to decolonization. While science and technology can bring exciting developments in health, food, and the understanding of our natural and physical world, these systems can also be used for extreme destruction. Science, imperialism, and militarism have been entangled at the technological level, but also at an ideological level which seeks to establish science as an apolitical activity. Democratizing science entails acknowledging the politics of science and for scientists to step into a peacemaking role. We invite our audience to think of the way science and technology have played a role in the current crises around the world. We urge Concordia to completely divest and cut ties with weapons manufacturers such as Lockheed Martin. And previously, Concordia has successfully divested from fossil fuels, and this precedent can lay a successful path to similar avenues. We hope you keep these ideas close to you as we explore the different facets of SynBio democratization and that you carry them with you into your work beyond this space.
Thanks, Zoe. So today we welcome Dr. Matteo Farinella. Again, thank you for joining us this morning. He is a scientific multimedia producer at the University Columbia Zuckerman Institute. Matteo has a PhD in neuroscience from University College London, and since then he's been combining his scientific expertise with his lifelong passion of drawing to make science more clear and accessible to a wider public. Through the Art Meets Science event, Matteo will be presenting his story as well as guiding us through a workshop on his creative process to translate complicated research into a tangible illustration that can be used to better convey your research or even just a difficult to understand scientific topic of your choosing. So this workshop is to encourage researchers, especially in the field of synthetic biology or any science, to create a picture of the downstream application of your project that can truly capture its importance and potential impact on humanity. So, yeah. So I guess we'll just start.
No, I'm good. Yeah, thank you for having me. And thank you for the people joining us online. Unfortunately, I cannot see you, but yeah, Zoe and Kathleen are gonna help me interact with you during the more practical parts. So let me get my pointer. Sorry, too fast. Okay. So yeah, I'm gonna tell you a bit more about my work and how I do what I do. I'm going to structure this like I'm going to give you a bit of theory first. I'm going to tell you about visual thinking and storytelling, some of the things we touched upon yesterday. And then we're going to get into some practical exercises to start thinking how to visualize your science. Even if you're not skilled at drawing, you may not want to do the final figures, but I think it's very useful for really anybody to just try to summarize your research visually. So I'm going to get started. Just a bit more about myself. I work, as Kathleen mentioned, at Columbia Zuckerman Institute, which is the latest new neuroscience institute at Columbia University. It does a lot of different aspects. It works on a lot of different aspects of neuroscience. It publishes on all the best neuroscience journals. And our job, I always have to specify, it's not just me. I'm part of the communication team there. And our job is to take all this exciting, high-profile research and turn into something a bit more accessible for non-scientists. Because unfortunately, the way it's written and presented is not, let's say, it's not always the most engaging. And in my case, this often takes the form of visuals. I do many kind of different visuals, from more traditional diagrams, graphic abstract, which is maybe what you're more familiar with and what you want to work on today, to more creative illustrations, like for journal covers or press releases. And then when I really have more time to play and experiment, we like to do some animations, kind of basic explainers about neuroscience. But these I'm happy to talk more about any of these things, but yeah, animation definitely take much more time. So we're not going to get to that today. And how did I get to do this? I have a background in neuroscience. I studied cellular molecular biology as an undergrad. Then I went on to study neurobiology, and my PhD was actually in computational neuroscience. So not even a particularly visual part of neuroscience. But throughout these years, I always had this passion for comics. I grew up reading comics, not really superhero American comics, but more kind of the Italian equivalent of this, like serial comics from France and Italy. There's a strong comic culture there. And yeah, I love to draw. I've been drawing since I can hold a pencil, and I love to tell stories. So over the years, I got more and more serious about this, and I started making my own comics. But for most of the time, like really up until the end of my PhD, I kept these two things very separate. I was trying to be a good science student during the day, and I kept my comics for my evenings and weekends. And my comics were not about science. It seems kind of crazy now that for so many years I didn't even think about combining the two. But yeah, I was drawing the comics I was reading, so it was more like autobiographical, fantasy, weird, underground comics. This changed at the end of my PhD when I met Hannah Ross, who you will see she's then the co-author on my book. And I always had to give her credit because Hannah was really the first one from my scientific community that when she learned about my passion for comics, she didn't just dismiss it as this fun fact, this like weird hobby I had, but she was really excited. She thought this could be a very useful thing for us to do, to communicate our science into comic format. So Anna, was a postdoc in the lab, so she was a scientist like me, but she also had a background in science communication. Specifically, she was making science documentaries. And science communication was this word that I was vaguely aware of, but I associated mostly with museums or, again, documentaries or scientific writing. So not really anything I was doing. But Anna, again, to her credit, she really pushed me and she said, no, this could work. We should do a comic about neuroscience. And that's how the idea of making neurocomic, our book, started. And our pitch was, we also applied for a grant to do this. And our pitch in a nutshell was that it's precisely because comics are not really considered fine art. They're definitely not associated with science, but they also don't carry the same weight and baggage of fine art, which, let's be honest, for non-artists, like as a scientist, as someone who never studied art, I think would be just as intimidating as science for non-scientists. Like these two worlds, I feel like they grew very apart and they don't often talk to each other. But again, comics, nobody has ever been scared of comics. We literally read them as children. Nobody ever say comics are too complicated for me. So when it comes to science communication, what is usually considered a baggage of comics, like that people don't take them very seriously, that are childish thing, when it comes to science communication, could actually be a useful tool. Again, because it could help us reach a whole new audience. So yeah, this is how the idea was born, and we were very lucky. We got a grant from the Wellcome Trust, which, in case you don't know them, they are this big funding body in the UK. Sorry, this was all back in London. That's why I did my PhD. And the Wellcome Trust supports a lot of basic research. In fact, my whole lab was funded by the Wellcome Trust. My PI was funded by the Wellcome Trust. But they also have a beautiful museum, the Wellcome Collection. If you happen to be in London, I highly recommend it, about the history of science. And they also gave these small grants for kind of new original science communication projects. So we got the grant to do this comic, and that's when it became really something real. And I had never really done a book before. Again, I didn't have any background as a writer, as an artist. I was really completely self-taught. But very early on, we decided that we didn't want to do a textbook in comic format because we already have textbook about neuroscience, and I think they're pretty good at what they're doing. If you want to learn everything about neuroscience, get the Candel textbook, and everything is there. And also, I think that's not the best thing comics can do. Comics are really good at telling stories. So we came up with this idea of having a character that is literally transported inside the brain and finds himself slightly lost and confused in this other world that initially looks like a forest. Then it will turn out that what look like trees are actually neurons, brain cells, and this forest is the brain. And it's going to meet all kind of weird characters, famous scientists. And as you can see, the comic is as action-driven as possible. It's not your traditional science book. And whatever the reason, I don't know if it was the storytelling or the nice cover design that the publisher made, but the comic was very successful. It came out in 2013, and it was quickly translated. I think it's now more than 10 languages, so thousands of copies. Like, I'm not trying to be modest. This was like way beyond our wildest dreams. In fact, in the grant application, our proposal was to just print a few hundred copies and kind of give them away at science festivals. And instead, it became this like international bestseller. So this kind of really kind of got my attention, woke me up to this idea of doing science communication through comics, which I never thought it would be a job. And in fact, it wasn't at the time, but encouraged by the success of Neurocomic, I left research. I mean, I finished my PhD around at the same time, so it was a good timing for me because I knew I didn't want to stay in research, but I didn't really know what other skills I had. And it turned out this was a skill, so I went freelance. I published a few other books, and yeah, just through my work, more and more scientists and scientific institutions started to get in touch with me. And I started working as an illustrator or science cartoonist. And that was great, but to complicate the story a bit further, because I have this academic background, I was really curious to know, like, why was I doing what I was doing? Like, were comics actually helping? Were people getting anything out of this? And if so, why? How do comics work? So I got very lucky because at the time I moved to New York, and Columbia University had this new program in society and neuroscience. And I got this 3-year scholarship to really do a deep dive and research this topic. So I essentially read everything I could about not just comics, but science visualization, science education, how visuals can help. And I tried to come up with some kind of not rules, but like some highlights, like why I think comics can be useful. And so this is what I'm gonna try to talk about today, give you the highlights. And the first question I had to answer, which it may seem obvious, but like I was never really taught about this throughout my scientific career, is what even are science graphics? What is a science graphic? There are many different kinds of science graphics, and it turns out there's a lot of books written about this. It's kind of a field of research in itself. I brought a couple here if you're curious. But yeah, just to give you a quick answer, yeah, I definitely recommend this last one, Building Science Graphics, by Jan Christiansen, who's the art editor of Scientific American. And this is a really like step-by-step guide to create these graphics. And one of the things she points out very early on, also in one of her blogs, is that there is a whole spectrum. You can have more figurative illustrations, like zoological reconstructions. You can have science diagrams. And you can get more and more abstract with these diagrams, up until scientific visualization, which is probably what you're most used to in your work. But all these things are science graphics. And even within one domain, one field, illustrator like Mary O'Reilly points out, you can represent the same things in very different ways, like DNA or a protein. Like you can go from the very realistic to the completely abstract. So there are a lot of choices that we make as scientists in these things, but we rarely think about them, and we're not trained to plan ahead what kind of graphics we want to use. So they end up being a bit of a mix, which is not necessarily a bad thing, actually. This, I think, is a nice example, a vintage example, where you can really mix like completely abstract visualization with very detailed, like ornate illustrations. And I think it works well. So this is something I want to keep in mind. You don't have to just pick one format or one solution. It's very interesting when you mix different things. Here's another example, again, from Jan Christiansen blog, how here, like this was a very technical math article about angles and rotations, and they added these little human characters to kind of like make the rotation more real, more relatable. So yeah, there are plenty of examples of these online. This is another very nice one from the project DR Data. I don't know if you have heard of this. These were two designers, information designer, infographic designers, who started sending each other completely hand-drawn postcards, trying to visualize things in their life. So, like from like different scents they smell throughout the week, or like I don't remember what is this, but like there were something about like how many doors you go through, like how many cups of coffee you had every day at what time. So it was a really nice way to kind of play with data visualization. And as I think you can see from my work, I also like to play a lot with the different styles. So that's another way in which I think I'm different from like maybe traditional artists, like where you have to have your own style, this like very recognizable style. I do everything from like very kind of realistic graphic novel style illustration to diagrams to completely abstract or cartoony characters. I don't really mind. I enjoy it all. Like my goal is to make science more interesting, more accessible. So my first question is usually like, who's my audience? Of course, I use different strategies, whether I'm talking to adults with a scientific backgrounds or young people. And then based on that, yeah, like you can move along the spectrum from more figurative to more abstract. So now, since we are not that many, I would like go around, like I want to know who you are, if you're okay, just giving a quick intro, like just your name, pronouns, and what you study in, what kind of graphics you're looking to do today, if you're interested in doing some graphics. Since I told you so much about myself. And I don't know, if anybody online wants to introduce themselves, feel free to write in the chat.
Yeah, I'm Zoe. My pronouns are she, her. I am a PhD in biology, and I'm looking at targeted drug delivery using ultrasound-mediated microbubble. And that is a very big mouthful, and I can never really explain it to parents, friends, family. So I think my audience would be, yeah, just folks in my everyday life. Yeah.
Okay, that's interesting.
Hi, my name is Bing Huang. My background is in chemical engineering, but right now my current interest is in neuroscience and AI. And I don't have any graphics project at the moment.
We'll find something.
I'm Michelle, she, her. I study in computation arts, which is already like art and science combined. I'm also part of the iGEM Concordia team. I'm making their wiki, so I hope this workshop will help me with that.
That's great.
Hi, I'm Julia. My pronouns are she, her. I'm studying lysosomes in the context of aging and neurodegenerative disorders. And so, yeah, I'm looking to try to explain my project better.
That's great.
Hello, I'm Kathleen. My pronouns are she, her. I'm studying extracellular vesicles, which are basically like bubbles that are created by cells to communicate with each other. And so I'm just trying to make a visual to explain my project on that.
My name's Zachary Valente, and my pronouns are he, him. I'm studying journalism. It's my third year. And I'm honestly just here today to capture the visual aspects of what this event has to offer.
My name's Hugh, he, him, and I'm a motion designer, and I came here to, me and Annika, who's here on my left, we're working on a project that's more in, kind of similar to this, but more in the realm of law. And so I'm kind of gathering inspiration for that project.
I want to hear more about that, but yeah, later.
Hi, I'm Annika, she, her. I have a minor in cognitive science, and I'm looking to make more graphics. I've been getting more into animation and digital art, so I thought it would be cool for informational graphics for cog sci things, linguistics, neuroscience.
All right. Okay. Thank you all. All right. So yeah, now we can start with the more practical tools, strategies. And the first thing I want to discuss is visual thinking, which we touched upon yesterday. But yeah, I really came to think of drawing not just as a final product that we are trying to get to, but more as a tool for scientists and people in general to structure their thoughts. And again, not the one of the things that visual can do, of course, is to help you stand out. But again, I want to go beyond that. I feel like most people, that's why they want visuals, because social medias, of course, they perform better. The algorithm literally favor posts with visuals. But that's a very superficial use of visual. I hope today we can go a bit deeper. And as I was saying, there are many authors that have written about this. Temple Grandin is always somebody I like to quote because, yeah, they are engineers, but also neurodiversity advocates, and they talk a lot about these two modalities: the visual modality and the verbal modality. And I don't know. I don't really think it's so clear cut. I think we both, we all use both, maybe in different ways. But they argue that that some people are really more visual thinkers. And that's especially useful in engineering and science because visual thinking helps you to make connections. So words are very linear, like you have to read one word at a time in a certain order, while visuals, you can see them all at once. And this helps you maybe to like look at things from different angles, different perspectives, that it maybe would be harder to do with writing. And I think this is especially important in comics because you can really like, again, there's a spectrum, like you can cover the spectrum from the completely linear, from like the classic 3 panels comic strip that you find in newspapers, to, you could argue even a completely non-linear like map or infographic is like a comic. This is taken from an article I wrote with some information designers, which was explicitly looking at comics for patterns for infographics. And yeah, I think the other beautiful thing is that you can choose. So you can combine them within the same story. You can go from a completely linear explanation to a completely linear, a non-linear splash page. Just to give you a few examples, the drawing is mine, but this quote is from Nick Souzanis, who's a famous comic scholar, and he really talks about this dual nature of comics. So you can read a story panel by panel, and in that sense it is like text. But also within a page, within the same comic spread, you can start to notice this like non-linear interaction across panels. And I personally think that this is very useful in science because the things we have to explain rarely fit in like 1, 2, 3, 4 linear structure. There are always exceptions, there are different scales to consider. So this is why I started to argue for comics, because I think they can do this very well. And again, just to give you a couple of examples, I don't know if we can have this a bit bigger. But yeah, this is a comic from Nick Souzanis himself, published in Nature. It's about climate change. But you can see how in the same page, like very seamlessly, he uses there's like a very linear, it starts very linearly. It's almost like a science diagram explaining the greenhouse effect. But then it starts to like break up the design a little bit. And there's this diagonal, especially, that, sorry, you don't even notice it. What have I done? Sorry. Let me get back to it. So this diagonal links the very abstract data of CO2 emissions with the very real effects of forest fires and desertification. So these are the kind of interesting layouts you can find in comics if you pay attention. Is there another example from this physics book by Johnson, The Dialogues? And here you can see how it's trying to explain how 2 forces, B and E, kind of affect each other. And it's kind of a loop. It doesn't even matter the science, but you can, this is another interesting kind of solution where you can keep going this loop forever. It's non-linear layouts. Okay, and this brings me to the first exercise I want to do with you, which I mentioned briefly yesterday. It's an exercise designed by Souzanis, who is also a comic educator. And I really like it because it takes off the pressure of for people that don't feel like drawing or they're not very skilled at drawing. So the prompt is you actually don't have to don't draw things. You can take a sheet of paper, you carve it up into panels or like shapes that represent the shape of your day. And then you fill it with marks and lines that represent your either physical or emotional activity throughout the day. So again, don't use words, try not to use symbols or realistic drawings. Just try to go completely abstract. Like think of it almost as a data visualization for your day. And it can be your regular day, or I don't know, it can be yesterday. Like, it's kind of a fun way, I think, to start thinking about layouts before getting into drawings. And we can take, how much they say? I think 5 minutes, like, let's do this quick. It's just a warm-up. Let me put the timer. And yeah, if anybody needs pens or colors, I have some stuff here, but I see you brought something. Thank you. It's hard already, or we haven't even started? I mean, if you're really stuck, he says I shouldn't show anything, just let you free, but here are some examples of the kind of things people do. So yeah, as you can say, it's basically like a map or an infographic of your day. This one actually breaks the rule because there are some notes, but those are just in your head. It's always fun to see what people do. I love the explanations. No, I mean, only if you want. But actually, it's fun to look at other people's ones and see what happened there. Yeah. I mean, the goal is really like you start to see how much, hopefully you see like how much you can already express without even using like figurative drawings or words. Like you can really, yeah, like there are a lot of things you can show just with colors or shapes and lines. And I think that's all part of visual thinking. And
Huh? Right brain? I feel like it's both.
it's always both.
Two hemisphere interconnection.
Easy for you to just like draw out on like day looks like very like technically like here you're like lines and shapes.
Yeah, well, but because you're good at drawing, but like for people that don't draw,
I know, but yeah.
I feel like this is like an easier way in. All right, how much we have left? 10 seconds. All right. So I'm curious. How was this? Did you find it? Did you find it useful? That's true. No, you could have done yesterday. Anybody wants to share, or like, should we just move on? Also, I don't know. Anybody online has any questions or anything you want to add? I hope. Yeah, usually it's nice to share, but if there are people online, people in person, I don't want to favor too much. All right, so let me move on. Like the next thing, I'm going to get more into storytelling, which is the title of the workshop, and we talked about it yesterday as well. Storytelling. So this video doesn't work, but I don't know. I'm curious. How many of you has anyone seen this before? I can just tell you a bit about it. It's actually an experiment, psychology experiment, very old one from 1944 by Heider and Simmel. And essentially, in the videos, there's really not much to see. There's no sound or nothing. It's just these abstract shapes moving around and kind of interacting with each other. And the experiment was simply just showing people this thing and just ask them, what did you see? And the correct answer is, of course, just abstract shapes moving around on the screen. But most people, I don't remember the statistics now, but like most people saw this like very violent confrontation between the two triangles trying to defend the circle. And it's fascinating because it shows, it was taken as evidence that storytelling, this thing we usually associate with entertainment and art, it's really something much deeper. It's a way for humans to understand the world. Really, that's the base of all like, what are they called? Like animate religions. Like people always saw intention behind winds and waves and sun. Like we cannot help project human emotions and intentions onto inanimate objects. So again, this was the first inkling that there is something deeper about storytelling. And then there are many authors like Joseph Campbell that argue that there are actually these like almost universal patterns of stories. This is an illustration I made for Scientific American about an article that actually shows that if you reframe your life story with this structure, people report being more satisfied with their lives. So there is something we don't know what it is, but there is something really hardwired in our brain that looks for stories, looks for characters, and find it somehow pleasing, these patterns of stories. So when it comes to education, again, like this is important because I don't know why, but we relegate narrative to only entertainment. But I think it could be very useful when we talk about science as well. Unfortunately, most science texts are expository, which again is not necessarily wrong. Expository texts are very good, but they assume there is a very motivated reader that is willing to sit with you and read the material. Narratives make things much easier to remember, they make things more engaging. And there are some psychologists that have been trying to study the impact of narratives. So that's why I started, sorry again, too fast. This is why I started to use more and more narrative in my comics. It's interesting because actually something I did, as I mentioned earlier, something I did almost intuitively with Neurocomic. We wrote a story about the brain, and then later on I found there is kind of scientific evidence for it. The problem with storytelling is that you need characters. And in science, it's often hard to figure out who's that character. That's why most scientific writing, scientific reporting is about medicine or biology, because it's much easier to find the character there. It's the patient or it's the doctor trying to cure the disease. But when we go to what I think of the two extremes, the very, very large or the very, very small, then it's hard to tell a story. Unless, again, you use comics and you can turn anything into a character. And I know this is something kind of frowned upon by some scientists, like we are taught not to anthropomorphize our subject and not to it's not, we wouldn't stick googly eyes on ourselves when we write a scientific paper. But again, I'm arguing for this only when it comes to science communication. When we are trying to talk to non-scientists, I think these things that comics do so naturally could actually be very useful. So again, think about your audience. If you're doing something for scientists, then definitely avoid this. But if you're doing something for children or for non-scientists, they could really help. And again, I think comics have a sort of special power here, because as comic scholars like Scott McCloud pointed out, comics often use these very simplified characters. Like some of the most popular characters in comics are essentially emojis, or they're not humans at all. And it's interesting because it's not like cartoonists cannot draw any better. So McCloud argues this is a implicit or explicit strategy to make this character more relatable. Because if I tell you the story of this one guy, as good as the story is, it's a very specific story. If I tell you the same story with this or this character, then anybody virtually can relate to that character and project themselves into that world. So again, this is something I find very often in science comics. Larry Gonick is definitely a pioneer of this. He did all these cartoon guides to biology and genetics, and you can turn anything like a repressor and operator into characters, like kind of fighting with each other to get to the DNA. This is something I grew up with. I wonder if it was popular here. It's a French animation. It was very popular in Italy as well in the 80s and 90s. And I think it's still a masterpiece. This is essentially the story of the human body with all the cells, like white blood cells and bacteria and viruses and blood cells all turned into characters. It's really good. I think I learned more biology from this than from my degree. And of course, not surprisingly, is something I do a lot in my own work. So yeah, my children books are all about these neuron characters that live in the brain city and do different jobs in different departments. I often turn bacteria into characters. And something else you can do, similar but a bit different, is to scale down humans. So put the humans in the domain of the cells of biology. And again, this may seem silly, but some people argue that it provides this kind of embodied understanding. Because the problem with science is often it takes us to these domains where most people don't have any experience. We never see a cell, we never touch a cell. So we don't, it's even hard to get a sense of scale or motion. But if you put the character in it, then it becomes a bit more real, a bit more relatable. So in summary, yeah, visuals, I think they help us reach beyond the human scale. So the human scale is very limited. Our experience goes from millimeters to maybe kilometers before cars. And yeah, we just think in terms of minutes or years or months, but science often asks us to think about deep space and deep time. And as I try to show here, I think comics can really bridge all these different things very conveniently. All right, so now, yeah, we are about halfway through, and I want to start getting into the more practical part. with these ideas in mind, these like visual thinking and storytelling. So let's start thinking about, I use a graphic abstract as an example because it's probably the graphic that most scientists are asked to do, but it doesn't have to be a graphic abstract. Like if you have your own idea, your own project for visual, like you can work on whatever you want. I think that the strategies are similar. So first of all, what is even a graphic abstract? As defined by Elsevier, it's a single, concise, pictorial visual summary of the main findings. So again, it doesn't have to summarize your whole research. It's more like, what are you studying, what do you do, and what do you find out? And it should be easy to understand. Like it shouldn't require a legend or too much of an explanation. And yeah, they give a few other advice. I don't find them particularly useful, but I guess I reported them there. Something I find useful, and again, I can send you the link, is this paper by Hallman and Bach, who, again, they're information designer, and they looked at as many graphic abstracts as they could find from different publications, and they tried to find common patterns. And they said that probably most authors, again, we don't have any training as a scientist, so most authors arrive at these patterns without really thinking about them, like why they're doing what they're doing. But they try to summarize them in the hope that it can become a bit more clear. Again, this one I could use a bit bigger. So first of all, they identify some dimensions. So the first thing you can think about is the layout, which is kind of what I was thinking about during the visual thinking session. So think spatially, like there's the most obvious solution, which is linear, but there are many other things you can do. Then there's depiction of time, text, and representation. But the layout, I think, is the most interesting. So here are a few, just like zoom in a bit more, these are a few examples they find. And again, they say like you can use arrows to connect the different parts, but ideally, I think you shouldn't even need arrows if the reading order is clear. But here are a few examples of like, this is a zigzag layout, which is very much like a comic, like three rows. Here in the West, we understand is from left to right, top to bottom. It's pretty easy. Another useful layout, especially if you are comparing different conditions or control versus experiment, is the forking path. Another one, again, for comparison, is parallel. And you can have parallel both horizontally or vertically. Again, I haven't read this paper, so I don't know what they're about, but like, you get a sense of like what I'm trying to show you. Here is also an example of nesting. So the classic like zoom in panel, people use it a lot. And this is also an example of orthogonal. So you can read it, this is interesting because you can read it both left from right, but also top to bottom. So it's really like almost, you can choose the reading order. Other useful thing to point out are different ways to depict time. So there is, you can use space, you can you actually break up the page like we just did in our exercise into boxes. You can use numbers, of course, and if it's not clear, you can add arrows or symbolic. I guess you can use sun to represent the day, and moon for the night. They also give statistics, how often people use these things. This picture didn't quite make it, but this was just an example of linear, left to right, kind of evolution. Sorry, I don't know why they're all pixelated. It's an example of arrows. You get the idea, I hope. And I brought some books so you can see some example of these things. But okay, the first major exercise I want to do with you is to start sketching your graphic, whatever that is. So again, the first thing I want you to think about your goal, like who are you trying to reach with this graphic? And also what are you trying to do? Are you trying to explain a basic concept? Are you describing an experiment you're doing? You're comparing different conditions? Like I would pick at most one or two, because again, we're trying to do something simple, something we can work together here within the hour. So yes, pick like a clear goal. And then we can start focusing on the image. So like once you know what you're doing, you can even write it out if you want, if you have a laptop or something. Like I usually start with writing it out, even my comics. So like 2 or 3 sentences. And then my advice is try to translate as much information as you can into visuals, and then try to move things around on the page. And then the most important is probably like revise. So we can either do it together or break up into groups maybe first. Just show it to each other and see how much the other person understands. So let me put a timer for this. And then we can regroup. I can walk around and talk to you, and then we can regroup and compare. And again, I don't know, I feel bad for the online people. Like if you want to do this with us, I don't know, you can do it. You can take a picture and send it to us. We can give you feedback.
Sure. For those who are online, you're more than welcome to turn on your cameras, unmute, and also show your work if you so choose to participate. We can see you here in the space if you want, so feel free to participate
Also,
that way.
anybody has any questions before I guess we jump into the practical part? All perfectly clear? All right, then start thinking about your graphic. And again, if you don't have, for the non-scientists, I don't know. I guess it could be, maybe I'm gonna match you with some scientists to give them feedback after, I don't know, you wanna, so we have 4 scientists and 3 non-scientists. I mean, do you have something you think you can work on? Okay. You know. All right. So get started, and then I'm gonna actually put a timer for 15 minutes, and then I'm gonna check in with you, and we can do some comparison, some feedback. And if you are really stuck, like, yeah, this is another structure that some people suggest is try to summarize into 4 panels and use this ABT formula. So something and something else, but there's like a problem, like some challenge, something you want to answer, and therefore you do something. I don't often use this, but if you find it useful, you can give it a try. want to see some books, I'm going to put them over here. This is really good, like in terms of like plenty of different examples. These are all interviews with like science visualization designers.
Trying to make things right. I've got good music on my radio. I can hear love songs playing from door to door. I don't want no one to come in to catch me side. But ever since you left, said goodbye, I wish you were here with me. I wish you were here with me. Thinking about your kiss, no more around me. It seems like love is never gonna find me here. I keep on looking for a brighter day, but all I see is time slipping away. I've got to stop, get myself together. All I can see is stormy weather. Wish you were here with me. I wish you were here with I've got to stop, get myself together, 'cause all I can see is stormy weather. Wish you were here with me. I wish you were here. Wish you were here. Oh, Lord, I just wish you were here. Come on and walk with me.
Seems like you've been wasting time trying to find your place in line. You need to get your ducks all in a row. Stop acting so peculiar. Get on board, I won't fool ya. Follow me, I'll show you what you don't know. So take some time to understand that life is a race for each and every man. Sometimes people will treat you like a worn out shoe, but they don't know that you can't lose. Now if you seem to get off track, I'll be there to get you back. Don't you worry, I'll be in your corner. When the chips are stacked against you, just remember that I'm with you. Keep on moving right past the finish line. So take some time to understand that life is a race for each and every man. Sometimes people will treat you like worn out shoe, but they don't know that you can't lose. Come and be a winner. Come and be a winner. Come and be a winner. Be a winner. Slow winner. Be a winner now. Now you're a winner. Oh, baby. Oh, now you're a winner. You're a winner. Yeah, yes, you are. Now you're a winner. Ooh, because you got me. Now you're a winner. Ooh, because you got me. Yeah. Now you're a winner. Because you got me. Uh-huh. Now you're a winner. Ooh, because you got me. Now you're a winner.
My Sherry, you're more lovely as a summer day. My Sherry, you're more distant as the Milky Way. My Sherry, you're more pretty little one that I adore. You're the only girl my heart beats for. I wish that you were mine. In a cafe, or sometimes on a crowded street. I've been near you, but you never noticed me. My Sherry, oh Lord, won't you tell me how could you ignore that behind that little smile I wore? I wish that you were mine. La 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 la. La 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 la. Maybe someday you'll see my face among the crowd.
on scientists, pair with scientists. It's ultimate. But because, yeah, like you probably know each other's work already, so. So I don't know what's the best way to do. You can swap seats, or like, I don't know, the three of you can come in front here as you prefer. We can just make 2 groups. We are odd numbers, so. I mean, you have your own. All right, can I get, I don't know, maybe.
Maybe the three of you just like move.
Yes.
And then they'll just be like us because we're all science. Well, you're science too.
I mean, it's a pretty small group, so we can also all do it all together. Again, I'm sorry for the online people. Like, feel free to send us your pictures if you want. But like, who wants to start showing us their diagram? And the idea is like, yeah, no, that's my timer. The idea is to like see how we can improve it. Yes. Okay. Yeah. You have something?
Okay, so I don't really have a research project, but I decided to explain the concept of how synapses fire from one neuron to the next. So I know that there's like this famous quote, I can't remember who it's by, but it's by a scientist, who says that the dendrites are like the receptacle cells. Oh, just a blonde, thank you. That the recepting neuron and the delivering neuron, they don't ever touch. They just kiss in the way that French ladies don't want to ruin their powder, so they go like.
I didn't know that quote.
And they never actually touch. So I've drawn lips on all the dendrites, and then I zoom in, and they go mwah mwah, and they're giving it to the next one.
I love it. I never heard that before, but it's.
So that's that.
Yeah. Okay.
How could I make it better?
Yeah, I mean, it's pretty good already.
Maybe if you could like give them little faces and make them into little French ladies.
Yeah, I think
that would be
good. I think that'd be good. Yeah, it also doesn't show that much who is the recepting neuron and who is the delivering neuron.
Yeah, that's the main problem. Like, yeah, it's unidirectional, so it's only from axons to dendrites. So that's one. I didn't even talk about metaphors. Usually it's one of the, like, I love metaphors. I could talk about metaphors for hours, but yeah, it's hard to find good metaphor that like really map on your concept. So in this case, yeah, it would be just one kissing the other.
Yeah, I was thinking maybe it's like one of the ladies has a cold and she gives the cold, even if she's not touching, she gives the cold by like breathing on to her, like she gives like the germs to the next neuron.
Yeah, I mean, yeah, I don't know if I can figure it out now, how to.
I like the kissing thing. I think it could work with just like one looking kind of bashful.
Yeah.
And not like only receiving the kisses and the other.
Yeah.
Mwah, mwah.
Also, the makeup element is cute. We can see them put on makeup
powder. Yeah, that'd be cute.
But anyway, the main concept, yeah, is true that like there's no actual connection, touching. It's all. And then the other thing, yeah, would be like showing like something pass between the two. Chemical messages.
Make something pass. Okay, I'll work on that. Thank you.
But the layout, I think, is pretty good. They use the the zoom in version.
Thank you. Who wants to go next?
So, my thing has to do with etymology, and I found out recently that the etymology of the word addiction comes from the word in Greek adicheri, which means religious devotion. And I thought that was kind of spot on. So I was thinking of an animation where we have an image of a church that morphs into a syringe. So this top of the church would kind of morph into the end of the syringe. Sorry? Oh.
I love how you are all kind of going for metaphors. I didn't even mention metaphors, but like.
I thought that was the idea.
No, I mean, I use a lot of metaphors in my work. So that's one I didn't know.
can really pick the church steeple that looks like a syringe. Like,
Yeah, did you make it up?
It's very easy the way that it makes sense in my head.
All right. What about the scientists? Who want to start?
Like mine is so technical now. Like
Well, but that's why we are here together.
I think
right?
I need to find a metaphor for mine. Okay, just ignore the giant text because I was just trying to like, yeah. So I mean, my research is on these extracellular vesicles that are being produced by these producers. So like the whole idea is that like cells communicate, and the way that they do that is by these vesicles that they produce, and these vesicles can contain like molecular information, and once that vesicle goes to a different cell, then it can change like the physiological state of whatever cell it gets into, right? So say like, okay, there's like a cancer cell and you want to give it a specific drug in order to cure that cancer. So the idea is like, okay, like let's put that therapeutic drug into this vesicle so that it can get delivered and targeted. So I was trying to show that here by, so the first panel is like, sorry, you're gonna have to zoom in really. Like it's, yeah. The cells are producing all of these vesicles, which are these small little dots. And I did like a zoom in of what the dot contains. So in this situation, it would be that therapeutic molecule. And then here I have like an unhappy, like diseased cell. And then I'm showing like the arrow that's showing the vesicle that's going into the diseased cell. And then it goes through some like physiological repairing. And then you just have like a healthy cell at the end.
Yeah, that gets complicated.
That's complicated.
Let's see. I know. Any feedback before I? Any idea?
Feedback, please.
I like the healthy and disease cell.
Yeah, that's cute.
But every single time like you explain your project, you always say like, oh, vesicles are bubbles. So I think it would be fun if you start like
Saying bubbles.
yeah, like putting, I don't know, a bubble or like a cell releasing a bubble
Yeah.
in some type of way.
Maybe even when you were talking about like the disease cell and the normal cell, if we talked about anthropomorphizing, that's how that works. Like giving human characteristics to the individual cells, maybe one could be really happy and the other's kind of cranky.
Yeah. Yeah.
But it's also a matter of audience because this could really work for like an abstract.
Yeah.
Yeah, honestly, my main feedback here is like more about the layout, like that, and I know it's only like a first sketch, but like, yeah, like you're going from like the large scale to the small scale, to the large scale again, and then there's a before after. So I think the before after, it's definitely like I think the most important thing for you. So I would just like do a kind of split design. So like, you have the unhappy cell here, and the before, and the happy cell after. I'm just like, I don't know, I'm just thinking how we can like rearrange this.
Yeah, because that is like the main message, I guess. The
Yeah. So maybe it would be like something like this is really where you put the vesicles in. And maybe like, do you need to show the actual vesicle, like what's inside, or
I guess you don't. Like if it's already obvious in some way that.
Or how about we combine the two things? So like you have the vesicle and you have the tools inside, the repair tools inside.
Oh, true.
And then you can maybe have, I don't know, who makes this, who produced these vesicles?
The a different healthy cell. That makes sense.
So yeah, you can have like the vesicles going in and then you just zoom in. I don't quite know what goes here. Maybe it's to figure out, but yeah, like this would be easier to read for me.
Yeah.
Top to bottom,
Actually,
left to right.
yeah,
I mean, often like literally like this is the main advice I have for scientists, like move things around, but you have all the right ingredients. All right.
Thank you.
Who wants to go next?
Yes, I can go next. I guess I have to explain a bit my project to understand it. So here, what I was trying to do is like the steps of what I need to do in my project to get to the endpoint. And so basically, the whole point of my project is to tag a protein that's in lysosomes. Well, I'm using yeast, so bacillus. And this tag, what it does is whatever proteins come into close proximity is going to tag it with a different thing, and then I can know whatever is interacting with this protein. So starting my project, I have to do CRISPR. So I guess cutting the gene and then inserting that tag. So I try to do that with like cutting and then like pasting it with the tag. And then this is where it gets a bit more technical. So in order for the tag to work, you need to introduce biotin. So that's what I'm trying to say here.
And what is biotin? I guess you need to explain that somehow.
Biotin is just like whatever the tag uses to tag the other proteins.
Okay.
Is that a chemical or?
I'm not sure. But I guess it's kind of like
It's what makes it visible.
Yeah.
That has been
it's
cut.
It's a tag. It's like ubiquitin. So it's a tag, and so yeah. So here, like I wanted to zoom in and show like I don't know what's around my protein, but once the biotin is in there, like they can be known with the tag. I know it's hard, but
This is quite complicated.
yes, I know.
So the end goal is to
Identify the proteins
okay.
that are interacting with the main one. It's hard. Yes, it's complicated.
And what cells are these? Like, why are these protein interested? I guess that maybe I'm missing like the opening question. Like, why are you looking for these proteins?
Well, in the long run, is that this specific proteins are involved in fusion of lysosomes. So it is believed that these proteins stop working or are involved in like a process that causes neurodegeneration or aging. So
Okay.
I want to see
The proteins
the
that
proteins
that
are
is.
involved in.
I see. Okay, so maybe like that's the part that is missing for me at the moment. Like setting up the problem. It's like, so we know these proteins are involved in fusing lysosomes, and we know that in neurodegeneration, this process goes wrong.
So give it more.
Therefore, you're starting from the technical part.
Okay.
So again, it's probably just a matter of rearranging things. Can't quite figure it out now, but again, like I'm happy to stay. We have more time, so like if you want to try again.
Okay.
All right. Do you want to go? We go in order.
Sure. I'm not a scientist. Well,
Okay. Sorry, I bunched you with a scientist.
not in a way that matters today. Like I'm a computer scientist, but
Okay.
I'm in a bio project.
Oh, that's good.
So our project this year is bioremediation. I don't know how to explain it again. I don't know much. I'm just making the website that I'm trying to understand so I can like visualize it better. So we're making yeast that binds to nickel. So we're gonna introduce that into water to help remove the nickel.
So nickel is bad. That's the first thing you want to explain. Yeah.
So I guess it comes from like water that comes from mines.
Yeah.
So we want to remove the nickel from the water. And we're creating yeast that binds
Okay.
to it.
Okay.
So I created like a 4-panel here. It starts with there's nickel in the water. and then you zoom in. I don't know how to draw yeast, but
Yeah, they are pretty much just little bubbles. So
the word yeast will do it. We introduce that, and then the yeast and the nickel binds. I guess I also don't really know how that happens, but I'm more about, learning how to do the visuals, and then I'll learn how the process actually happens later. So it binds together, and then you zoom out in the last panel, and wow, there's no more nickel in the water.
no, that's a pretty linear story, so it's easy. Again, I think the main thing that we all forget as experts is like to explain like why are we doing something. So like, yeah, why are you looking for this protein? Like, why is nickel bad? I guess I vaguely know, but like I don't really like know. So I mean, a very easy addition here would be like, I don't know, I guess like fish, like that fish, like here, and then like the fish is happy here. Like it's like these like very like simple like visual cues, like so people understand like this is bad, this is good.
Also, like how you morph the word yeast into just like the compound of yeast. That's,
Yeah.
Because it's like the title becomes the actual element of the drawings.
That was kind of
Crossover.
on accident because I didn't know how to draw yeast, and I was like, I guess it looks like a what?
And also just like a minor note, but like, yeah, the way you, again, I mean, I don't know, I kind of like it visually that you like swap the two, but maybe it's a bit confusing. Like I would just keep it like a 2x2 simple square. So like, and maybe it's like macro and micro. So like you have before and after, and yeah, you have the water here with the dead fishes and the nickel. I don't know, I'm making it up. Does it actually, is bad for fish? I assume it's bad, but, and then again, you can zoom in and like show, okay, we introduce this yeast. And also you can, yeah, this is where like color is very useful. You can just show the nickel like before is in the water, it's outside, and then it like goes inside the yeast or whatever it is, or like it's digested. But like, yeah, it's pretty linear. Again, just moving things around and keeping things simple. Okay. Zoe?
Are you from the iGym team? Sorry.
Yeah.
Oh, I'm a mentor.
Okay.
So yeah, it's really cool that you're coming here.
I guess I don't know what to do.
Thank you. So here's my guy. So my project is about targeted drug delivery, and so that means, conventional drug delivery techniques. You take whatever drug you have, and it just goes through your whole body. So I drew this guy, and he has like a ribbon going through his whole body, and that's going to be the bloodstream. I want my first panel to be like, this is how it usually goes, and have every single organ affected, so maybe like just shining in green or something. And then say, but if we can localize it to one single place with bubbles, and when I think of bubbles, I think of like soap bubbles and people blowing bubbles. So I put a little guy blowing bubbles into the bloodstream. Then we can open up the bloodstream and deliver drugs in that specific context, and that's going to be like third panel. Again, maybe all of the organs, and one of them is encased in a bubble, and only that one has the drug in it, and the other ones are just like fine.
Yeah, makes sense. This is one of, I mean, you say also this is meant not really for scientists, but more like for children. So yeah, I mean, I love the draw. You really went for the creative drawing, which I guess in this case is fine. Yeah, if it was for like, yeah, that's kind of what I struggle all the time as well. Like I enjoy drawing, so I want to make them super detailed. I think we touched on this yesterday. The question is like, when do the art get in the way of the science? So just make sure not to add too much detail.
Yeah.
Otherwise, it may get confusing.
Yeah.
But definitely, yeah, using colors to show that the drug. And again, maybe like the part that is missing is like, why is the drug going everywhere bad? I guess we know that it's bad because there are all kind of side effects, but maybe that's not obvious for non-experts.
How do you use the interactions between, because a comic form is also a multimedia form, right? Where you have text complementing images. How do you decide to prioritize? Like, would this be a case where I can stick it in this first panel and say, if all of these organs are affected, then we have side effects?
Yeah, I mean, also, so what kind of drugs are these? What do they?
This is a drug delivery platform, so it can be used for
any. It doesn't really
any.
matter. Okay.
But chemotherapy, for example.
Yeah, I was thinking, yeah, of like the specific example. I mean, of course, we can come up with like metaphors. Classic one is like, yeah, drugs as like a carpet bombing, of course, is not good. Like you want to be more targeted. But yeah, it's hard with cancer because you're not targeting some specific bacteria.
Maybe you could do like a metaphor of a garden and there's like a weed in the garden.
Oh,
You're not
yeah.
going to put weed killer all over the garden. You're going to put it on that space. Much nicer than carpet bombing.
Yeah, I know. I never liked the militaristic metaphors. Unfortunately, yeah, that's the first one we use for.
Is that the term they use though? Carpet bombing?
Yeah.
Referring to like hitting a drug to the whole body?
Like.
No, that's not a medical term. But yeah, like that's the problem. We usually like. I mean, antibiotics, for example, like you have to like you kill also your own like bacteria, good bacteria. Instead of like if you have targeted drugs, you kill only the things you need to kill. Yeah, cancer a bit more complicated, but yeah, weed could be good.
I could make like the organs have grass and then.
Yeah, maybe it's a garden.
Yeah.
And like the tulip is like a liver and stuff, and it's like you're looking for a specific thing. You don't want to.
That's a very nice visual. I mean, there's a point where like just like brambles and like thorny, like all tangled up, and that's the thing you're trying to get to. I don't know if the bubble works anymore with that, but you can try.
Yeah. Maybe the spray will be like coming out in bubbles or something.
Yeah, you can keep the bubbles. Okay, that was good. We have anything online? Always checking. I don't know if there's still anybody with us or they gave up on us. I mean, I had a few examples. Can I get the slides back? Like there are some like from Cell actually, like they show you before after examples, before the redesign, so you can see like it's very similar to the thing we're talking about. Like this is before some abstract that somebody submitted, and I don't know what do you think is wrong with this? Let's see, what would you change without even knowing about the topic? Any suggestions? Yeah?
It's very confusing. I don't know where to look.
I mean, they did put arrows, but yeah, like first of all, it's like the reading order is very confusing. I think that's the first thing they changed actually. So you see like go clearly top to bottom. And yeah, like it's also confusing because like apart from the reading order, like then you go here, but actually this one is meant to be read top to bottom. So it's like a forking path. So again, keep it all in line and then fork. Yeah, this is what they say. Reorienting the elements, which is like the main feedback I had for like in most cases. Also the colors have been adjusted to highlight in the right focus. Before, yeah, they were a bit all over the place. It was not clear what we were supposed to look at. Another example is this one. What do you find confusing about this one? I think it's mostly colors. Like there are, there are way too many colors, so they cleaned it up, and yeah, this before-after thing that many of you have, like, make it very clear. Here, I don't even know where it was supposed to be. I didn't even notice before there was a before-after, and here is very clear. Reoriented, some arrow, color palette. Yeah, it's good to always have like a focal point. So it's like either before, after, or yeah, like make very clear, like nickel is bad, so we want to remove it. Like, and again, color can be very useful for these things. Is there anything else? Okay, yeah, I add a list of like resources if you want to keep working. Again, we have 20 minutes left. Like, you're welcome to stay. We can keep working on yours if you want. I'm here. If you want to keep working them at home, I'm going to give you a quick like list of things that you can use, again, to finish them, because the idea here was more to structure it, and then of course you have to like do it digitally and like color it and all that kind of stuff. So very quickly, yeah, these are a lot of similar things. Like the graphic abstract hospital is like what we just did, the kind of before after. These are just the references. There is also this visual co-working session, which I attended a few times. It's kind of nice. It's like different illustrators, designers, different levels, just working together on Zoom, and then you can ask feedback. And if you're struggling with something, you have people like me that can give you feedback. More concrete examples. Practical things to consider. I mean, maybe this is boring for the visual people, but like vectors versus raster, most scientists don't really appreciate the difference. Vectors are scalable, so if you zoom in, if you have to make a poster for a conference, they scale very well. Raster formats, they don't, so that they end up pixelated. Color is also important. You should use RGB for screen images, things mostly for web. CMYK is for printing. These are all settings you can choose in Photoshop or Illustrator, whatever you're using. Also, picking color is very important. This is a very nice tool called Brewer, where you can try different scales, and you can also make sure they are colorblind safe, print friendly. You can toggle on and off all these parameters, and then you just like, get the 3 colors and do your own figures on this. This is mostly meant for maps, but if you don't know where to start, it's also a good resource. If you really don't want to draw yourself, there is a bunch of websites where you can get free icons. I think this, if I remember correctly, the Noun project, this is all Creative Commons, so you can use them as long as you give credit. I think each one has like its own, like it tells you how you should credit it, if you should credit it. This one I think is you have to pay, or maybe there's a free plan, but if you then want to actually use them in papers, you have to pay a subscription, but it's more specific for biology. This is a similar thing for medicine, health-related icons. And again, I can share the presentation, so if you can explore them. BioRender, you probably encounter it, is becoming this very popular software to create scientific figures. Again, you can sign up for free, but then you have to pay to have the full access. But it's very good. They have like all different kinds of icons you may need. You can change colors. You can literally do the whole figure in there and then export it. It's another BioRender. Or if you, or probably your PI has funding, you can hire a science illustrator. There are associations where you can find names. There is the Medical Illustration Animation Association. There is GNCI, it's a what does it stand for? I forgot. I was member for a while. Is the Guild of Natural Science Illustrators. So it's more for like more detailed illustrations of animals or plants. And then this is new, but I wanted to include it because yeah, it's Biocreate Index created by Grace Chang, who's like she was the art director, I think, for Ginkgo. We heard yesterday from Danielle. So yeah, she worked with a lot of illustrators and artists over the years, and she just, I don't know, just for fun, she just put together this beautiful website where you can actually search bio artists, science illustrators, and it also tells you if people are available for work or not. I'm in there as well. And finally, yeah, I just wanted to give you my contacts if you want to follow up. As I said, we still have, I'm still here for 15 minutes if anybody wants to like work more on their abstract. Otherwise, especially people from home, feel free to get in touch, and like if you need feedback on something, I'm always happy to give a quick look. And thank you for playing with me.
I know that there are a few people on Zoom, so I'm just going to underline that again for you, Matteo. If anyone wants to jump in and add something to the chat or show a drawing or anything like that, feel free, and we can we'll help make that visible to everyone here.
People willing to give feedback.
Yeah.
Did you get enough?
No,
Yeah.
the garden.
Is this an event that like comes all day, or is it just?
No, I'm just visiting the, sorry, let me mute myself.
No big deal. Okay, everyone joining us on Zoom and in the live stream, just to let you know that we will be closing up the Zoom and live stream now, but the workshop remains available on our YouTube channel if you'd like to revisit or share it. And thank you so much for joining us. And don't forget to come back later on this afternoon for some more events related to the SynBio expressions. Thank you.